everyone, my name is Cedra, and for my protein project today we have MC1R, which stands for melanocortin 1 receptor, and it is a GPCR or G protein coupled receptor found on the plasma membrane of melanocytes, which are specialized skin cells meant to keep the skin safe and is responsible for pigment production. So it plays a crucial role in melanin synthesis and determines skin and hair color and protects against UV radiation. So mutations in MC1R are associated with fair skin, red hair, and an increased risk of melanoma or skin cancer. So that is the disease associated with it, unfortunately. And it is due to a reduced production of eumelanin, which is a dark pigment that absorbs UV rays. And instead, morphomelanin, which is a lighter pigment with weaker UV protection, is produced, increasing susceptibility to sunburn and DNA damage. So I'm going to reverse the camera and we're going to get into the structure of the 3D protein. So MC1R has a 7 transmembrane domain structure, and what this means, it weaves through the cell 7 times. So this shape is common among GPCRs and is essential for receiving signals from the environment. And the extracellular region, so that would be at the top portion of the protein, it binds to melanocyte stimulating hormones, or MSH, and the intracellular region activates signaling pathways. So to represent this, I have a 3D model printed using beads to represent the amino acids, because MC1R actually has 317 amino acids, so I used a bunch of different beads to represent that. And then you have the lovely bedazzling to show the helical structure. Moving on into the mutations, so this is the normal unmutated MC1R protein. As you can see here at position 151, we have arginine, and then in the mutated structure, we have cysteine. And for how it relates to their function, arginine is positively charged, so it can form ionic bonds and hydrogen bonds, and these interactions help MC1R maintain its shape and function by stabilizing the structure. Meanwhile, cysteine is neutral and is not charged, as it has a thiol or SH functional group, and it interacts differently with other molecules, and this weakens MC1R's ability to interact with MSH, making the receptor less effective at sending signals. The name of this mutation is R151C, and we talked about how the structure impacts the protein, and it's the changing of another one amino acid for another, and it makes MC1R less effective at activating CAMP signaling, which in turn reduces eumelanin production and protection against UV radiation. And for how these mutations can further impact organisms rather than just disease, people with this mutation tend to have red hair or red or blonde hair, fair skin, and freckles. And this is because their skin does not block UV radiation as well, which creates a higher risk of sunburn and melanoma. The last thing I want to cover on the structure of MC1R is the different levels of protein structure. So for MC1R specifically, we have the primary originally, which is the linear amino acid sequence. So like I said earlier, this protein has 317 amino acids. So it's 7 TM domains, and this concludes of a string of letters and phosphorylation sites. And for secondary, we have the local folding and the A helixes and then the B turns forming these loops. For tertiary, we have the 3D structure of the single polypeptide. And this is just a compact GPCR structure with ligand binding and signaling regions, just to ensure all the messages are being shared from the protein. And for quaternary, the quaternary structure of MC1R can form dimers or oligomers in living cells. So it's just its interaction with other proteins, as MC1R is a single subunit protein. I hope I was able to teach you so much about the MC1R protein, and that you learned how the MC1R protein impacts organisms, how structure impacts function, as well as what changes about the molecule and its impact when the mutation happens, when the mutation occurs. The last thing I want to address is some limitations in my project, starting with of the size and scale. So this model is definitely not to scale, as proteins are much smaller and dynamic and much harder to see with the human eye. This also goes into my material representation, as it does not show any bond types or flexible movements that affect function as it actually does in the protein. And then for the simplification, the intracellular signaling pathways are much more complex than I'm able to show with a 3D model, but I hope I was able to create a fun and visual piece to learn more about the MC1R protein and how it works. Lastly, for the limitation, the chains also continue, so it can connect and signal to other proteins, so they would continue on here after it passes through the membrane 7 times, but I unfortunately did not have enough material to represent that. But I hope you learned a lot about MC1R and liked my model.